studies. My name is Marcia Castro. I'm the Endelot Professor of Demography and Chair of the Department of Global Health and Population at the Harvard T.H. Chan School of Public Health, and I'm going to be the moderator of this seminar. I'm pleased to introduce our speaker today, Dr. Vicente Vérez Bencomo, Director of the Finlay Institute of Vaccines in Cuba, who is going to discuss the COVID-19 vaccine development and deployment in Cuba. Cuba may be the first Latin American country to develop and manufacture its own vaccine against COVID-19. It's the only country in the region that has not joined COVAX, but has 5 vaccines in development. And some of those have reached final stages of phase 3 and now are under intervention. Just this week, last Monday, Cuba began a massive COVID-19 immunization of its pediatric population with one of the vaccines, Soberana 2, and became the first country to vaccinate over 2 years old population. By the end of September, by the end of this month, it's likely that all Cuban citizens would have received at least one dose of those vaccines. So before I hand the floor and we start hearing about the Cuban case, I want to just go over very quickly some housekeeping items. First, we're recording this seminar and it's going to be available on the David Rockefeller Center YouTube channel, but also on the Global Health and Population website shortly after this session, and we're going to email everyone who registered for the event the link to the seminar. Second, we hope that you're also going to join us in other lectures and seminars and series that we host here at GHP, at Global Health, but also at the David Rockefeller Center. And the last thing I want to mention is that the chat function is disabled, but we welcome your questions. So please feel free to send your questions through the Q&A tab at the bottom of your screen. We're going to hold the questions to the end, but feel free to submit your questions at any time. So, Jimena, do we have the presentation?
Yes, I'm ready to share the presentation.
Okay, so then without further ado, I want to welcome our speaker, Dr. Vicente Vérez Bencomo. Thank you so much for joining us, and we look forward to hear the Cuban example on the vaccine development. Dr. Vérez?
Thank you very much, Marcia, for this invitation. That's a big honor for us. We are here, not only myself, but part of the team that developed this. I need before the introduction, I need to explain that here in Cuba, we have 2 main institutions developing vaccines. One is the Finlay Vaccine Institute that developed a series of Soberanas, and there is another institution developed also their own vaccines that are also in use in Cuba. So I will concentrate myself on the series of vaccine of Soberana, and then I will try to explain this year and 2 months, because this is 14-month stories. This very accelerated development of this vaccine that are now in use in our country. So before starting, this is a collaboration with several institutions, among them the University of Havana that contribute in some way, especially the Faculty of Chemistry, and also the Center of Molecular Immunology and our Finlay Vaccine Institute. So may I have the first slide, please? Okay, so the rational design and development of this vaccine was based on several concepts because we are not new in the vaccine field. Our institution is working on the vaccine field for more than 30 years, and most of the vaccine that we are using in Cuba came from our institution. Our institution is organized in a way to move from the bench to the market. It means that we are organizing in order to do everything, including basic science. So the rational design of this vaccine was recently published as a review in ACA, American Chemical Society Central Science. So for those interested they can get there. One of the main concepts, may I have the next slide, please? No, those before. Oh, no.
Another one?
Two okay, another one before that. No. No?
Got it.
Okay.
This one? Okay, sorry about that.
Yeah, thank you. So one of the main ideas, the first idea, because as you know, it was accelerated development. So everything that usually you do during 5-6 years, here we need to do in 2-3 months. So this accelerated development make us to make a very quick decision and challenging decisions sometimes. So that's why the first one I want to just to explain was the antigen selection. So we decide to select a protein, a small protein that is called receptor binding domain from spike protein. We decide to produce this protein in mammalian cells because we wanted to have mammalian glycosylation as in order to mimic the protein in the virus, because in the virus, in humans, the protein is glycosylated by a human glycosylated machinery. And then also another important principle is the multi-epitope presentation. So we try to express this protein in several copies in a different alternative of vaccine that we decide. And the last one, in order associated with the antigen selection, was the adequate, that we make everything in order to have adequate orientation for a maximum receptor binding motif exposure. Receptor binding motif is the part of the protein that interact with the human ACA2 receptors. So that's critical in order to enter the human cells and in order to induce infection following by multiplication of the virus. So the general idea was to induce antibodies, so induce immune system to respond to this protein, and then to elaborate antibodies and other type of immune response in order to avoid, to stop this interaction of the virus with the cell, in order to avoid the entrance of the virus in the cell. The next slide, please. May I have the next slides?
Yeah, sorry, it's stuck, so I'm trying to, sorry, just.
Okay.
Okay, so the next one is right here.
Is this one?
Yes.
Okay. Okay. The second important decision was the priming strategy, because in order to wake up the immune system, you need to really send signal that there is something wrong coming up. So that's why we decide to the priming strategy to be based on existing platform at the Finlay Vaccine Institute. So there are two platforms. The first one is to make conjugate vaccine, because we developed several conjugate vaccines that are now in use. So that's why we decide, in the case of Soberana 02, we decide to make a conjugate vaccine of this receptor binding domain, a small protein, and tetanus toxoid. And the main idea was to involve T-cell cooperation evoked by T-cell epitope of tetanus toxoid. And the second vaccine, Soberana 01, we tried to bring innate immunity involvement, and this we try to make that just by including Neisseria meningitidis, other membrane vesicle, that is a very well-known innate immune stimulator. The next slide, please. Okay, and then we develop also a booster strategy. And then, because after priming, either by natural immunity, in the case of convalescent, or by priming with other vaccine, we believe that there is a need, and we found during our development, we found that there is a need on a possibility of a booster response. So that's why, in the case there is a strong memory response there, so then booster can just increase the antibody response in order to avoid the virus to multiply. So that's why we use, in the case of the booster strategy, we use the same protein in endomeric form, and then we develop what we call Soberana Plus, that is the third vaccine that we are under development. And then in this particular case, we try to make antigen presentation without intervention of other immunoactive component, and just the receptor binding domain in this dimeric form, looking at the very selected booster response with minimal side effect. Next, please.
I'm gonna change it. It's stuck.
Okay.
I'm sorry.
Okay.
I'm sorry.
No.
There we go. Yes.
Okay. So overall, there are three among the 5 vaccines that we are developing here in Cuba. There are the 3 vaccines that we developed. I just summarize. Soberana 1 is based on innate immunity stimulation by Neisseria meningitidis, all the membrane vesicles. The Soberanas 02 is a conjugate vaccine to tetanus toxoid in order to evoke a T-cell response and then T-cell memory. And Soberana Plus is just a recall of this specific immunity against the receptor binding domain. May I have the next, please? Okay, so most of these ideas are summarized in some of our papers. Next, please. And then I need to explain that there is a lot of basic science there. And but there also a huge industrial biotechnological operation in order to be able, for example, to produce a vaccine like Soberana 02, because there is a center of molecular immunology producing the receptor binding domain in CHO cells. There, then the Finlay Vaccine Institute produced tetanus toxoid in our facilities. We have also additional facilities for making the chemical conjugation of this receptor binding domain with the tetanus toxoid, and then formulation and filling is performing in another institution that has facilities for that. Next, please. And then I would like to explain the first combination of the 2 vaccines, in Soberana 02 and Soberana Plus. We established a clinical trial development plan consisting of the phase 1 that we start early in October last year. Then phase 2 means that this divided in phase 2A and phase 2B. In the phase 2A, we just confirm the result of the phase 1, and then we move to phase 2B, and then phase 3, and then stepwise studies, and then phase 2 in children, phase 1 and 2 in children. So that was the general plan of the clinical trial. Next, please. Yes, and then after phase 1 and phase 2A, we decide that the combination of vaccine that we will further develop in this line was a combination of 3 doses, means 2 doses of Soberana 02 for priming, and 1 dose of Soberana Plus for boosting of this response. And this was the combination that we further developed in this clinical trial. Next, please. Okay, I will just pick some slides. Of course, there are a lot of material that are under preparation to be published. There are at least 7 or 8 papers in preparation now, but I just pick some slide in order to show you and in order to illustrate what we get from that. And then you can see there, the next, please. You can see the immune response. It means antibodies against the receptor binding domain induced by immunization with 2 doses of Soberana 02. It means 2 doses of the vaccine conjugated to tetanus toxoid. You can see there that there is around more than 60% of people responded in what we consider is a threshold value, minimal value for IgG response. And then after the 3rd dose, next please, we see that there is an important increase, over 90% of people attending this value. So that was the main idea we get from all the pool analysis of phase 1, phase 2. And with this enhanced, next please. Of course, we demonstrated that the quality of these antibodies and the ability of these antibodies to neutralize the live virus is at least equal to a convalescent panel, the Cuban convalescent panel seros. Next, please. And then also we demonstrated another important issue with this, that we expect from this conjugate vaccine is the induction of T-cell response. We proved that in some of these studies, with the small studies with a number of people, we proved that there is really the effect of T-cell response is induced by just 2 doses of RNA2. It means that the priming is not only the result of the priming with the 2 doses of the conjugate vaccine is not only the antibodies, it's also memory. And its memory is T-cell memory, it's B-cell memory, and this means this capacity that after a booster response, the capacity of increasing dramatically the antibody response. So that was the main result of this from phase 2 to phase 2 trial. And then with this enhanced, next please. We apply and we get a permission in last March, we get a permission to enter, sorry, this is a shift, this is not this one, it's the previous one. We get the permission to enter, no, I mean the slide. This is the wrong. There is the previous one is showing that we get the permission to move to a phase 3. With the phase 3 with 44,000 people, in March, and then next, please, next slide. And with this enhanced, we enter phase 3 that takes several months and end by the end of July, beginning of August. And in this phase 3, I will show you the interim report, but the final report is very close to this interim report that we apply for getting emergency use authorization. And you can see there that there is a really a very important one dose has some efficacy already for a severe disease. 2 doses increased to an important level over 60%, and then 3 doses has a 90.2% efficacy versus placebo. This was really very nice results that, next please, that also show that there are some efficacy in preventing infection. It was something expected. In the design of the vaccine, it means that the conjugate vaccine in the case of bacterial antigens show that really there is an important impact on not only on the disease, but also on infection. And then it was what we were expecting here also. And then we found that there is over 70% efficacy in preventing infection also. And of course, a very high efficacy in preventing severe disease and death. Next, please. With these results, we apply to our national regulatory authorities, and then we get emergency use authorization on the 20th of August this year. Next, please. With this enhanced, we get the permission in June when already the result we're moving forward. We get the permission to start clinical trial in children. I need to explain a strategy in children because there is a big difference with our vaccine and some of the vaccines that are now being tested in children, like for example the Pfizer, Moderna, or others. In our case, we are basing the platform that is used normally in children. So it means tetanus toxoid conjugate vaccines are used in infants size 2 months old for Haemophilus influenza type B, for Streptococcus pneumonia, for meningococcal, of course, for bacteria. But here are more or less the same platform, so we expect more or less the same safety. So we are moving in a very well-known platform. So that's why our strategy, because we discuss internally in the country, and we consider that given the safety, the proof safety of this platform of vaccine, and given also the ethical issues, we didn't consider that certainly a placebo double-blind control efficacy trial in children was really possible, because certainly with the pandemic in place, we consider that there is not sense to use placebo in children. So that's why our strategy, because first of the proven safety, our strategy was to correlate, to try to perform a phase 1/2 with enough sample, and then to correlate the immune response with the immune response obtaining in adults in the clinical trial for efficacy. And this is what we did. And look what happened. Next, please. We were very surprised to see that if you look at the 74% of people getting the immune response over 8, that this it was the threshold that we established on the efficacy trial. And in the case of young people, teenage, in the case of teenage, we get 93% of teenagers attaining this interval, dramatically more than what we get in adults. And in case of smaller children from 2 to 11, we get even better results. Almost every child responds to the vaccine, to the 2 doses of vaccine, and then certainly I will not show, but 3 third dose increase even higher the number, the immune response of the children and teenagers. And then with this, next please. Of course, the quality of the antibodies was also very high and comparable with a very positive comparison with the adults. Next, please. With this enhanced, we consider that we get all the information in order to extend the emergency use authorization for children. And after a long review from our national regulatory authorities, we get permission to use for the emergency use authorization in children. This was, in our opinion, a very important milestone, because that's true that children, maybe would you consider that children are not really very prone to serious disease? But anyway, in Cuba, we have some children dying also, and some children getting the serious disease. This is one problem. And the second problem is certainly that children are an important space for a virus to multiply. So certainly we believe that vaccinating children, we will be able to increase the coverage of vaccinated people and then to move closer to the point where there is really a very important community immunity established. So that's why we get this permission, and we start mass vaccination of our children from 2 to 11 since last Sunday, and then today we have already several tens of thousands of children immunized. I need to stress that the vaccine is confirmed that it's very safe, really, in children. There is no important adverse event in children, and then consider the high immune response in them. Certainly, in my opinion, there is an important action that we are performing there. Next, please. Anyway, I will be able to respond to any question about the children immunization. Additional development of Soberana Plus. I remind you that Soberana Plus is the vaccine that is just based on the receptor binding domain dimer without any other immunoactive compounds. It's just made in order to boost existing immune response. And that's why we decide, okay, can we be able to boost the immune response in convalescent? Next, please. The idea was, okay, in the case of convalescent, you get the virus, you become infected or sick, and then you produce antibodies or the immune response again a lot of potential antigens in the virus. And then when we just add receptor binding domain at the booster, then what we did in fact is just to select these clones of the B cells specifically that responds against the receptor binding domain that are known to be high neutralizing. So that's why the general idea is that with this, maybe we are able to improve the quality of the immune response of the convalescent, the quality and also the quantity. Next, please. So that's why we embark ourselves in a series of clinical trials with COVID-19 convalescent. The first one was a phase 1 clinical trial that was started very early. Really, this idea came to us very early, more than several months ago, and then we performed phase 1 with 30 convalescents. And this is now accepted and will be published soon in Lancet Regional Health, the Americas. And then after successful phase 1, we decided to move to a phase 2 clinical trial. And this phase 2 was a placebo-controlled clinical trial. And then overall, these 2 clinical trials were made in conjunction in order to be able to ask for emergency use authorization for this type of person. Next, please. And we get a very interesting results. Look at the T0 are convalescent that naturally has immune response, and on the right corner there is a typical panel of Cuban convalescent, and what we get is that around 80% of people move from the original immune response to a higher immune response. And with this, next please. With this enhanced, we come to a very, in my opinion, this is a very interesting result. This is a very new finding. This is just some days ago because we are applying now for emergency use authorization. Look at what we found. We found that in the case, next please, in order to, okay. So look at the 100% of convalescent from the moderate disease. All these patients respond with a very important increase in antibody response. The people having mild disease, they are 86% of those people increase dramatically their immune response. And even those that are considered asymptomatic people, even those, more than 50% of those increase their immunity. This is a very clear picture that shows that asymptomatic spectra of the COVID-19, in this spectra, some of the people has not really very strong immune response, didn't develop a very strong memory immune response. But in the case of the other, those that are considered, I consider really COVID-19 cases, mild and moderate, a very high proportion of those people, it has a very dramatic increase in their antibody levels. Next, please. And look at that, look at this study. We, in the collaboration with the hospital in Italy, this is a hospital, Amadeo di Savoia, we study live virus neutralization of the serum of convalescent. And this is neutralization of the serum of convalescent before immunization with a single dose of this Soberana Plus. Look at the left corner, there is a neutralization of the normal strain of the virus, and then the middle is the alpha, and on the right corner there is a delta that is the most serious problem that we have faced now. And look at that. Most of convalescent have neutralization for the original virus, but only a few ones have neutralization of the live virus against the delta. So there is, in our opinion, a very high risk of reinfection of those convalescent with the delta variant. So that's why that's why the vaccination, a single vaccination with a single dose of this type of vaccine of convalescent has a lot of sense now. So that's why, next please. That's why we apply a single dose and look at what happened after 4 weeks. Next please. There is an important increase in the antibody response that we have seen with the original one, similar to Alpha. But what happened with Delta? Next, please. With Delta, we see that almost all the convalescent now neutralize the Delta variant. So that's a very important finding, because certainly there is a high risk of reinfection, in our opinion, and this high risk of reinfection can be avoided just by a single dose immunization of those convalescents. Next, please. Also, we found that there is an increase. It wasn't unexpected, because we expect that the original immune response has already T-cell response. But then we found that there is some specific T-cell response against the epitope in the receptor binding domain protein. So that's why we found that there is also an increase in the T-cell response after a single dose. Next, please. And with this enhanced, we are now applying for emergency use authorization extension for convalescent, for adult convalescent after for 18 years and on. So that's why we expect that in the next weeks or the next coming days we will receive this authorization, and we plan also to immunize the 500,000 of Cuban convalescents. Taking together the 500,000 of Cuban convalescents and the 2.2 million of children, we expect to increase the coverage of the vaccination in Cuba from around 70% to over 90% of people vaccinated. And in our opinion, this will have an impact. This will have an impact on the disease, is what we expect. So we will measure the effectiveness and efficacy of this action. Next, please. And of course, I want to go back to the first development. This is an additional development. In the case of Soberana 1, I remind you that this is the dimeric receptor binding domain in a combination. This is not a chemical conjugation, it's just a combination with Neisseria meningitidis of the membrane vesicle. And I remind you that this is a very strong innate immunity stimulator. Next, please. So we make a phase 1 with this in comparison with and without all the membrane vesicle, and we found certainly that there is an improvement in the immune response by the presence of the Neisseria meningitidis of the membrane vesicles. And another finding is that we found that really there is a better homogeneity in the people responding. So that's why we decide to move. Next, please. We decide to move to a phase 2/3. This is the head-to-head non-inferiority trial. And this inferiority trial that is performed in 2 municipalities of Cienfuegos province in Cuba. This is the central part of Cuba. And in this trial, we compare 2 doses of Soberana 2 with 2 doses of Soberana 1, and in both cases, 1 booster of Soberana Plus. So we want just to look at the impact on the innate immunity stimulant on that, and we are especially interested to see what happened, especially in Asian people, because certainly in Asian people, the innate immunity can play an important role. Next, please. We are also planning additional studies, because in the case you see that where the memory immune response is not strong enough, then the booster of Soberana Plus didn't function very well. So that's why we think that Soberana 1 can potentially be a better response in this particular case where memory response is weaker. So that's why we plan also to study this in that direction, and there is something that we are going on, and we plan to perform this study. Next, please. Okay, so that's what we have for you to present. This was a really 14 months, really very amazing story. We really enjoyed. We dedicated ourselves as never before in order to be able to do that in 14 months. And so that's why I wanted to be accompanied by the small group of our colleagues that certainly dedicated a lot of their time for that. So I'm open to any question.
Thank you, Dr. Vicente. That's really an incredible team effort, and we all congratulate what you've been doing and all that you're still going to accomplish. So we have several questions. I'm going to start with one question that I think it's important just to clarify a lot of misunderstanding that's going on. Can you talk to us a little bit about if and how all this development in Cuba has some collaboration with PAHO or with the WHO? And in elaborating this, to what extent the development could have been even faster if there were not all the different kinds of embargo on Cuba. So if you could talk a little bit about this collaboration with PAHO, WHO, and embargo.
Yes, let me explain that certainly we have strong collaboration with PAHO, our organization, in the development, in the usual development of the vaccine. That is our historical links between the PAHO and our institute. And certainly we appreciated a lot this collaboration. Let me explain what we are doing. We are certainly now working with the PAHO, our organization, in order to certainly apply at some moment for the pre-qualification of the vaccine, and also in order to supply all the information in order to be recognized by the World Health Organization. That's something that is now under development. But let me explain. To do that in 14 months, certainly you need to be free to take a decision every hour. You need to be free to do what you decide to do at every moment. And then we started collaboration at the beginning with the COVAX initiative, or some of the initiative. And certainly there was some discussion at some point. At some point, we applied for help in some way, but we found that certainly the pathway in our particular case, the pathway to get that was not in a time frame that we were expecting. So that's why we decided we didn't close the links. We keep the links, all the doors open, and we keep informed constantly about our development. But anyway, we understand in our particular case that we need to be based on our own capacity to do that, because otherwise the time frame was very short. The time frame was very short. The restriction you're talking about Cuba, the restrictions are very important. It means that there is a very difficult for us to have access to funds, especially if they are originated from US, and most of the funds usually are originated from the US in some way. So that's why we decide. We tried several times, but then we keep trying. But then we decide that the main strategy should be to be based on our own effort, and that's what I did.
And is there any plan to apply for emergency authorization at the WHO? Because basically Cuba started as being left out from COVAX, but you could be a major exporter of vaccines, at least to Latin America. So are there any plans around this?
Sure. We are now under discussion with the World Health Organization. We have an important part of the file, especially the one that is associated with all this clinical development is ready to discuss with the experts and authorities. We have one strategy is to publish in a high rank journal because this is an important reviewer recognition is one direction, and second direction is expert for the World Health Organization. We are starting discussion with them, and then we need to certainly to move forward in the coming weeks or months. Certainly, there is two different lines on that direction. One line is the clinical trial results. These clinical trial results were performed under a very high level standard. We are performing also clinical trial outside. So we have a phase 3 trial with Soberana 2 and Plus also that is conducted by the Pasteur Institute of Iran. Now in Iran, they are performing phase 1. We have a strong collaboration with them. This is a part of the Pasteur Institute network. And then we have a very strong collaboration with them. And then we expect to have a confirming phase 3 trial result with a very high level, a very high standard also. So certainly these results are open for discussion at any moment and with all the experts. So we welcome all the discussion. And the second one is that certainly you need to understand that for a poor country like Cuba, even with a very important biotech industry developed, but for a poor country like Cuba, under this pandemic situation, we use all our facilities to do that. And these are not always the high standard facilities that can be considered, I don't know, from Europe or for North America. So we just modify and use all the facilities we have because we have really in a very important emergency situation. And this means that certainly in order to be able to produce larger quantity of vaccine with a maybe a level acceptable from Europe and for North America, we need investment. We are performing this investment now, but this certainly needs a small time in order to bridge all these things. So certainly we are moving forward also with the production. We have a good production facility, means around 10 million dose per month. That is more than enough for Cuba and maybe will be a good contribution for some, especially some countries that didn't apply a single vaccine yet. But anyway, we have aspiration to be able to produce a higher number of doses of vaccine. So that's why we are not relying only on the COVID-19 vaccine, because certainly we are based on a platform. So this is a multi-use platform that we want to develop at the largest scale, because certainly we have a pneumococcal conjugate vaccine also very close to be approved. And so we need to produce that. And we have Haemophilus influenza type B vaccine that was the first vaccine with the synthetic antigen ever developed 20 years ago. So we are now on an investment for a multi-use capacity for producing, not only because COVID-19, okay, we can contribute a lot in the next couple of years, but certainly we need to have a capacity that will be useful, especially for the poorest countries. And that's why we plan this multi-use investment in order to be able to produce in the same facilities also pneumococcal vaccine or Haemophilus influenza type B or some of the combined vaccine that we use for children.
Great. So you mentioned that some of the resources you couldn't have access, Cuba couldn't have access, and it is a poor country. So can you talk to us a little bit about how Cuba was able to, you already mentioned the facilities, all the facilities were dedicated to this, but how was the country able to put the necessary resources, considering all the challenges and the many different pockets you could not reach? So how was Cuba able to do this and put the resources needed to do what you guys did?
You want for me the magic answer? I would say the first things and the most important You know what is education? We have a very well-trained and educated people, and with a very high commitment with what we are doing. So that's why we were able to ask for our researchers and our people. We were able to ask more than usually what they ask in other places. So certainly the secret was the personal, the people. It was the human beings that was working there. They were able to do a very high sacrifice during these months. And this I consider I can challenge any place in the world to try to do the same, because certainly those people were so dedicated with such a high motivation. And whenever we knocked a door was open all the time. Any door in Cuba were open when we just asked, because there was a massive and a national pride effort. So certainly, but did you ask me the magic formula? Education, not resource, not. We didn't have the billions that multinational companies. Also, you take this billion and you make you put zero there, and then billions on the zero are zero. So that's what we have. We have almost no resource. We just use what we have, and then we concentrated on doing that. And this, in my opinion, this is just a secret, of course.
Okay, so I hope that besides writing about the efficacy of the vaccine, you also write that as a case study on how to get things done. There are some questions here on the chat about the fact that Cuba received some vaccines from China. Can you talk a little bit about this? Did you guys receive vaccine from China? Are they being used at all? Or Cuba is only using the vaccines developed in the country?
Okay. I will answer this last question, and then I will try to my colleagues to say something, because otherwise they didn't work for their coffee. Okay, so yes, we were open to any donation, because certainly we have a difficult economical situation. In any case, we didn't have money enough to buy vaccines, so that's why we were open to receive donation of the vaccine. And then certainly we received some of the vaccine, and then we decided that we will apply this vaccine certainly in a combination with our vaccine. So there is some experience that I cannot disclose exactly now, because it will be a subject of further information, especially when we will discuss the results. But in fact, what we're trying to do is just, we can in some way mimic the natural immunity by the disease by inactive vaccine. So that's why we are trying just to use inactive vaccine in a combination with Soberana Plus, in order to get the same result we get with the convalescent, where we have a very high immune response, especially today, in the scenario of the Delta situation, where we have a very high transmission. So that's why what I must say is that we are open to any donation of vaccines, and then when we get the donation of vaccines, so we try to apply this vaccine in a combination in order to sort out immunity at least at the same level as we get with our own vaccine. You can notice that we decided in all our vaccine cases, we decided to use 3 doses already at the beginning, because our ideas was to get the highest immunity level possible, because we were really expecting some new mutants at some way. So that's why we, since the beginning, we believe that there is not the 2-dose a system that we were expecting. We're expecting, in any case, a 3-dose with the highest level possible immunity. So that's why any donation of vaccine that we receive, we are applying now, and we're using that. and this will help us in order to get this more than 90% coverage during September. We expect to have more than 90% coverage with 3 doses in the whole population of Cuba by the beginning of November.
Okay, so one of the things that are coming up now in studies of vaccine effectiveness is that some of the vaccines, for example, like the Sinovac, Coronavac, it has reduced efficacy for older people. So from the results you showed, it seems younger people had better results, and particularly, and this relates to one of the questions that was asked, why do you think that children had such a great response to the Soberana vaccines? And connected to that, would you expect that the third dose that you described will resolve any issues of reduced efficacy at older ages?
Okay. Our first approach was justly to give a priming immunization schedule for in a way of conjugate vaccine, just for activate immunological mechanisms of B and T cells. Because you know the conjugation of the RVD to the tetanus toxoid in person or in population that has previously been immunized with tetanus toxoid, because normally it's a vaccine that we use in our immunization schedule, it will have some advantage in terms of the activation of T cells response, and this cooperation between T cells and specific B cells against RVD, it could be important in terms of the induction of B cells memory. After that, we decide to administer a third dose with a different vaccines. We have the results in our clinical trials. We prove the third dose with one more dose of Soberana 02, and we prove the third dose with Soberana Plus. And we found that when we give the first dose with a different vaccine, with an heterologous presentation of the antigen, we got better results. For this reason, we focus our immunization schedule in heterologous immunization with 2 doses of conjugate vaccines for a best priming of the immune response, and then a booster dose with only with the RVD in the medical presentation, just for boost the immune response against the more neutralizing epitope in the molecular RVD. Also, as for the beginning, we defined that our immunization schedule, the timing between those will be 28 days. As we know, as much larger is the time between those, the immune response is better. That means that we decide for 28 days, because in pandemic, you cannot give the dose so longer. Usually in conjugate vaccines, the usual time between doses is 2 months. Here we use 1 month between doses, and we are getting excellent results. And even now we have the first result of the long lasting of the immune response. And we have found that the people immunized with this schedule, this heterologous schedule, they still have antibodies after 6 months of vaccination. That means that the priming and also the booster dose are able to induce a high level of antibody and also long-lasting immune response.
Okay, so I'm going to ask just one more question because we are running out of time, but, oh, sorry.
Sorry, I forgot. And when in the phase 2, when we compare the data, the immune response between young people and older people, actually we found that the young people has higher immune response than the older people, but we have excellent immune response in the older people. And in phase 3, when we calculate the clinical efficacy against symptomatic disease, of course, the number, the percent of efficacy in all age group is 91% after the 3 doses. But when you stratify the sample by age, we have around 77% of efficacy in older people and higher than 91 in young people. That means that even in older people where the immune system is more senescent in their fusion, we have excellent immune response in these people, and could be also related with the priming of the response that is able to induce with the conjugate vaccine.
Okay, we have lots of questions, but there's no way we can cover all of them. So we're out of time, but I want to finish with one question, which is, you are the pioneer in vaccinating children. Nobody else in the world is doing this. So 2-part question. The first one is, as the population is being vaccinated, are you also trying to follow up to be able to evaluate vaccine effectiveness as you vaccinate, do things alongside? And 2, any conversations with WHO at all? Have they approached Cuba to start some kind of agreement that the whole world can benefit from vaccines for children, considering that nobody else is doing this? And in some places, children are being severely affected. So I'd love your thoughts on this.
Okay, so concerning vaccination children, of course we will follow. So we are now organizing as we start by the Cienfuegos province, and this Cienfuegos province we organize in order to follow up and then to sort out effectiveness and impact at the same time. So that's a study that is already discussed with the Minister of Health authorities, and now, of course, we will be following. And certainly, I need to say a final word about vaccination of children. You know, the Finlay Vaccine Institute is the organization that before the COVID-19 was the organization that was developing vaccine for prevention of disease in children. So this is our duty. This is our pressure. This is the thing that we really enjoy more, because certainly to know that you can save the life of children is something that cannot be compared with anything else in your life. So of course, when we started this 14 months ago, certainly our main dream was to be able to vaccinate children, because certainly somebody can argue, okay, children has not so prone to have a severe disease. Children has not so prone to be to die from that. But for the family who lose one children, this is 100%. There is not a statistical there. For those families, they will never recover their life. So a children is the most important for the family and for the society. So that's why our main dream was to be able to come to children. It was since the beginning. That's why we decided to move in this platform. And the result that we get in children, better immune response than in adults, was something in some way expected. Because certainly children have some vaccination with tetanus. They already have T-cell response against T-cell epitope in tetanus. So we plan to benefit from this T-cell response for tetanus in a better response in children. So that was something that was planned in a way. And we are really happy to see that's what happened. And certainly we want to contribute as much as possible in that direction in order to vaccinate the children, because certainly we observe that with the Delta, we have a tendency to be worse in children and worse in the younger age. But despite that, if you just save one children's life, this deserves all your effort. And this was our main motivation.
Thank you so much. I mean, and thanks for saying that, because the loss of lives in this pandemic is unbearable, and those are not numbers. Those are lives and families and dreams that have been disrupted. So thank you so much for saying this. Big congratulations to you and your team for doing what you've done. Right now, I want to invite you again to come back in a few months. We want to continue to hear the progress you're making, and we want to give voice to the work that you're doing, because as you know, we don't hear much about what's happening in terms of development of vaccine in Cuba. We want to give voice to that. So I hope you're going to agree to come back and speak to us again in a few months, hopefully with terrific results of the vaccination in children and of the incredible effects that Cuba can show with the massive vaccination of the population. So congratulations. I apologize to all the questions we couldn't raise, but thank you again. And I am sure I'm gonna see you back in a few months to hear more about the great work you're doing. Congratulations to all your team. Thank you.
Thank you. Thank you. Thank you.
¿Cuánta gente se supone que yo la vea?